Welcome to my webinar with the title Recovery of Cell-Free DNA using the QIAsymphony Circulating DNA Kit. My name is Alexander Wolf and I'm the technical lead for the liquid biopsy portfolio in molecular diagnostics at QIAGEN. And I would like to give you a general overview about this scanning and very interesting topic of liquid biopsy, and will also focus on some data from the fully automated purification of cell-free DNA using the QIAsymphony instrument. Here's a regulatory disclaimer for this webinar. The applications presented here are for research use only, not for use in diagnostic procedures. What does liquid biopsy mean? It's a non-invasive or minimal invasive technology, in most cases a blood collection, for sampling and analysis of molecular signs as diagnostic and monitoring tool for diseases. It's in clear contrast to tissue biopsies and has the potential to complement or even replace the tissue samples. Liquid biopsy can be divided into three major domains. First, circulating tumor cells, floating through the bloodstream in very rare numbers. Therefore, most challenging to purify and concentrate, but with the potential to obtain the complete genetic information in the form of the genomic DNA, as well as all types of RNA from a living cell. The second domain are the exosomes. That means microvesicles shed into the body fluids and transport mainly the cell-free RNA fraction between cells. The third fraction is the circulating cell-free nucleic acids, mainly the DNA, and highlighted on this slide here by the red frame, which will be the focus of my talk today. The cell-free DNA is released from dying cells into the bloodstream. In more detail, it's the genomic DNA of a dead cell which is strongly degraded by DNases into short fragments with a half-life of several minutes in the bloodstream. Currently, liquid biopsy is on the way to transform our healthcare tremendously. Despite the already existing success of liquid biopsy, an example for non-invasive prenatal testing or oncology, the application is still at the beginning of its revolution. This slide summarizes several aspects which illustrates the major benefits of liquid biopsy, which should hopefully convince you of the future potential of this application. I will shortly emphasize a few ones. Compared to tissue biopsy, it's obviously safer and less painful for the patient. Therefore, the compliance of a patient to undergo a simple blood draw will increase the potential of an early detection and close monitoring of diseases. For physicians, a real-time treatment response monitoring will allow increased insights on disease progression in combination with the faster development of new therapy options. Pathologists will face a faster, more cost-efficient processing and will obtain in parallel the complete heterogeneity of a disease state, because a blood sample reflects a more homogeneous sample type compared to a biopsy section. Analysis of the origin of cell-free DNA extracted from a plasma sample revealed that most DNA molecules derive from hematopoietic cells, which is not surprising because it simply reflects the high likelihood that dying leukocytes release its DNA into its surrounding liquid. Next, it depends on the size, the cell metabolism, and blood circulation of an organ, how much cell-free DNA is found in the bloodstream, as indicated on the right figure by the lung, liver, or intestine. Based on this knowledge, this is also true for the fetal DNA, shown here on the left side. With the advanced stage of pregnancy, increased amounts of DNA from the placenta is released into the mother's blood, and more than 20% can be found before birth. The same can be detected for cancer patients. Tumor size, blood supply, but also the therapy status of a patient is reflected by the amount of circulating tumor DNA in the bloodstream. Interestingly, also the total cell-free DNA amount varies for cancer patients and often reflects the overall health or disease status. Based on these findings, the striking success of non-invasive prenatal testing in the last years can be easily explained, and non-invasive prenatal testing is an essential part of prenatal screening today. Here, the preanalytical and analytical procedures are well harmonized and validated, and will most likely largely substitute amniocentesis within the next few years, or at least will further reduce these invasive diagnostics, which may are only required after a positive test result from the liquid biopsy-based assay, focusing on monogenetic disorders or chromosomal aberrations. Also at the beginning, highly expensive and not reimbursed by the health insurance, hundreds of thousands of women decided for the blood-based test to avoid the invasive procedure with residual risk of fetal loss. And currently, in many countries, the healthcare systems consider or already pay for the non-invasive prenatal testing for risk pregnancies, and discussions are even ongoing to expand the test to each pregnant woman due to the highly convincing results of the companies who offer the liquid biopsy-based test. The next field, which is on the road to a routine application, is oncology. At the moment, the cell-free DNA-based analysis is often used complementary to the tissue biopsy for monitoring the therapy response and resistance. But the data reveal that the cell-free DNA analysis from a plasma sample has the potential to identify rare mutations often responsible for metastasis and therapy resistance much earlier than the biopsy or imaging methods like CT. This is indicated here in this figure by the richly colored circulating tumor DNA in the blood tube on the lower left side, which represents the complete heterogeneity of the tumor mutation. Few cell-free DNA molecules already comprising mutations responsible for primary and later on secondary resistance may be present in the first blood samples collected already at the beginning of a targeted therapy. In combination with the more frequent sampling, easily possible for a simple blood draw, a faster and more precise prognosis of a therapy response is possible. Therefore, I think in near future, a targeted therapy for cancer patients without an accompanying liquid biopsy-based test is hardly imaginable. Now I will switch to the challenges of the cell-free DNA workflow. This slide shall illustrate the importance of standardized workflows for cell-free DNA applications. The pre-analytical workflow starts with the blood collection and stabilization. The cell-free DNA of interest, in example the fetal or mutant DNA as in the examples before, is present in a large background of maternal or wild-type DNA. Therefore, it is of highest importance to stabilize the cell-free DNA fraction after blood collection and prevent, in parallel, the release of genomic DNA from leukocytes. A dilution of the target cell-free DNA with background DNA will negatively impact the sensitivity and specificity of the downstream assays. Based on this knowledge, usage of cell-free DNA stabilizing blood collection tubes is highly recommended because in many cases a transport of the blood collection tubes for several hours or even for several days from the physician's office to the diagnostic laboratory is not unusual. Only in large hospitals, where a short transportation time at cool conditions for 1 to 2 hours between blood collection and plasma generation can be guaranteed, the usage of EDTA tubes is a reliable option. Next, it should be defined a clear plasma generation procedure. An example, one low-speed spin to separate the plasma from the cell fraction, or alternatively a second spin to remove potential residues of larger cell or DNA fragments should be considered and carefully tested in context of the clinical assay outcome. At which temperature and how long shall the plasma be stored before it's subjected to cell-free DNA purification? Here as well, a standardized scheme is recommended to ensure a high reproducibility of the results. International standards for isolation of circulating cell-free DNA from plasma are available now, which support and guide users for the establishment of standardized workflows. The next step in the pre-analytical workflow is the extraction process. Here I would like to emphasize that the choice of the cell-free DNA extraction kit also plays a major role for the reliability of the assay result. Completely independent of the extraction efficiency between different kit providers, each chemistry will have its bias on the nature of the eluted DNA. An example, it may impact the size distribution, the integrity of the double-stranded DNA, or it may favor defined AT or GC-rich sequences in the genome. All of this factor may affect the assay result. Additionally, of course, the assay itself and the data analysis have to deal with the specific detection of the target cell-free DNA in a huge background of maternal or wild-type cell-free DNA. A major challenge of cell-free DNA analysis is the low concentration of DNA found in plasma and the subsequent analyte, which is transferred into the downstream assay. This is indeed a clear drawback compared to cells or tissue samples as sample material, where the DNA input into the downstream assay is a rarely limiting factor. But here we talk about 1 to 50 nanograms DNA per ml plasma. In most instances, only about 1 to 10 nanograms, resulting in only a few molecules as biomarkers present in a sample, because you have to consider that your target DNA, that means the fetal or mutant cell-free DNA, is only present a few percent in the background DNA. The second challenge is the highly fragmented DNA, while the extraction must focus on an efficient recovery of small DNA fragments below 200 base pairs. Due to this challenge, it becomes clear that the question on the slide here, 500 microliter plasma, is this enough, as often used for genomic DNA extraction, are not compatible with the required sensitivity of the downstream assays for cell-free DNA. This issue must be addressed by input volumes in a range of 2 to 4 ml usually, but even up to 10 ml or higher as requested by customers. In parallel, the cell-free DNA has to be extracted quantitatively from plasma and diluted in a low elution volume to concentrate the DNA. But even then, the low concentration of approximately 0.1 to 1 nanogram cell-free DNA per microliter analyte makes the quantification using spectrophotometric methods like nanodrop impossible. Likewise, quantification by analysis of size distribution profiles is usually not recommended due to the limited sensitivity of the assays. Real-time PCR or digital PCR are the preferred options for a reliable quantification. But even for the PCRs, a bias in the total quantification, an example due to the choice of the target or amplicon size, should be considered. That's why we often prefer only a relative quantification to a reference method or reference conditions to assess the extraction efficiency. Based on this information, the requirements for an ideal cfDNA isolation method can be extracted. These are a fast and straightforward procedure to address the increasing sample numbers in the laboratories. We are talking about a throughput of a few samples per day, up to several hundreds per day, even 1,000 samples per day is not unusual. An easy handling of multiple samples in parallel, addressed by a fully automated extraction process with limited hands-on time and tracking of each sample. An extraction process for different sample input volumes to address the individual needs of the assay sensitivity. Of course, an efficient extraction efficiency focusing on the small DNA fragments, to ensure the detection of the low abundant target genes in the subsequent assays, and analytes containing highly concentrated cell-free DNA in an excellent quality to be compatible to a broad range of downstream applications, which are usually NGS or digital PCR. QIAGEN offers several solutions for cell-free DNA extraction, and I would like to focus on the two most important ones: a manual extraction kit and an automated kit running on a dedicated instrument. The manual one, the QIAamp Circulating Nucleic Acid Kit, allows a flexible starting volume from 1 to 5 ml plasma to give the maximum detection sensitivity. To handle the large processing volume, tube extenders for the mini columns and a vacuum device is integrated in the workflow. The robust procedure helps to minimize sample-to-sample cross-contamination and increases user safety when handling potentially infectious samples. The simple procedure is suitable for simultaneous processing of up to 24 samples in less than 2 hours. A flexible elution volume of 50 to 150 microliters can be applied. If higher nucleic acid concentrations are required, the elution volume can be further reduced as low as 20 microliters. The next slides will focus on the automated cfDNA extraction, which is highlighted here on the slide with a red frame. Due to the strong increase in sample numbers and due to the need for standardized workflows, there is a high demand for an automated extraction of cell-free DNA. The QIAsymphony Circulating DNA Kit offers 2 sample input volumes, 2 ml and 4 ml, and ensures in parallel a throughput of 96 samples in 5 to 6 hours. Solution volume is 60 microliters. Compared to the manual kit, a complete new chemistry in beads was developed, dedicated for the cell-free DNA extraction. A design goal for the new kit development was an equivalent performance to the manual kit, which is assessed as a gold standard in the market. The automated cell-free DNA extraction kit runs on the QIAsymphony sample preparation instrument. The instrument is well established in the research and diagnostic field for many years. Manifold applications run on this platform, like genomic DNA extraction from whole blood, Barcode, or FFPE tissue samples, as well as extraction of DNA and RNA from different viruses and pathogens. Therefore, we decided to expand the existing portfolio of the instrument to the cell-free DNA application. The QIAsymphony SP unit can be used as standalone sample extraction unit or can be combined with an assay setup for fully automated preparation of a PCR. The instrument supports a flexible sample loading from 1 to 96 samples, split into 4 batches, each of 1 to 24 samples. For each batch, a sample carrier rack with up to 24 samples can be processed consecutively in one integrated run, resulting in a maximum of 96 samples. The 5th carrier rack is available for internal controls or in case of the circulating DNA application to provide the proteinase K volume. The instrument also allows a continuous sample loading for batches 1 to 4 with an individual protocol selection. Each sample runs through automatic barcode reading, uploading of the sample carrier racks to ensure traceability. Traceability and safety is also implemented for the pre-filled reagent cartridges, which contain the buffers, magnetic beads, and enzymes. For each application, a specific chemistry was developed and pre-filled into the cartridges before sealed to ensure a long shelf life of the kits. A seal set, which is part of each kit, allows the reuse of an opened reagent cartridge for several weeks and thereby ensures the flexibility in sample numbers per run. Each graph is barcode labeled individually and detected by the instrument before the run, which allows an easy use, safe handling, and a traceable documentation. This figure depicts the generic extraction workflow on the QIAsymphony. The bead-based nucleic acid extraction ensures high yields and recovery rates. The excellent purity of the analyte supports the compatibility and sensitivity of the downstream applications. On the left hand here of the figure, a sample, buffer, and beads are distributed in a reaction vessel. Then, using a magnetic rod protected by a rod cover, the beads are collected and bind quantitatively to the rod cover. Then the beads can be simply transferred into a new reaction well in which new buffer is already dispensed by the 4 pipetting channels of the instrument. The beads can be easily released into the liquid when the magnetic rod is separated from the rod cover as shown here on the right side. By up and down movement only of the rod cover, the beads are mixed homogeneously in the processing volume and thereby ensures the high efficiency during the bind, wash, and dilution step on the instrument. This slide shows the extraction workflow for the cell-free DNA application for the standard protocols for 2 ml and 4 ml sample input volume on the QIAsymphony. The 2 ml protocol starts with the binding step, which contains an aliquot of 2 ml sample volume, binding buffer, proteinase K for digestion of DNA protein complexes, and the magnetic beads. After binding of the DNA to the beads are subjected to 3 wash steps before the purified cell-free DNA is released into solution during the elution step. The new dedicated chemistry for cell-free DNA extraction combines many advantages. First, addition of only a low volume of binding buffer to the sample results in a reduced overall processing volume, which allows a higher throughput compared, for example, to a chirotropic silica-based extraction. The other advantage is that all steps occurred at room temperature and that the new beads shall reduce that volume during the elution step. Difference to the 4 ml protocol is indicated here by the dotted line. After separation of the beads from the first aliquot of 2 ml sample, the beads are transferred into a new vessel which contains the second aliquot of 2 ml sample, binding buffer, and proteinase K. That means that binding of DNA from the second 2 ml sample aliquot takes place to the same beads which already carries the cell-free DNA from the first sample aliquot. After binding of the cell-free DNA, the beads run through the 3 wash steps before dilution takes place. I hope that this scheme also illustrates how we could address high input volume protocols up to 10 ml. We simply run through consecutive binding steps in 2 ml sample aliquots using the same bead portion. Due to the very low concentration of cell-free DNA found in plasma and urine, saturation of beads with DNA is not a limiting factor. As a result of strong increasing requests for such high input volume protocols by customers, we currently are working on the release of a standard protocol for 6 ml, 8 ml, and 10 ml sample input volume in near future as well. On the next few slides, I would like to sensitize you to important aspects of the assessment analysis of cell-free DNA. This slide shows 2 size distribution profiles of cell-free DNA analyzed with an Agilent high-sensitivity chip. The upper one shows the profile of cell-free DNA extracted from 4 ml plasma in blue and from 10 ml plasma in red. The blue curve is already at the limit of sensitivity of the assay, which makes clear that the reliable quantification cannot be recommended. Even the blank line don't indicate a failed extraction, but may reflect the donor with a low concentration of cell-free DNA in plasma, which is then under the limit of sensitivity for this assay here. On the other hand, the red curve reveals a nice size profile with a major peak at approximately 170 base pairs, indicating the cell-free DNA fraction coiled around the histone complex and therefore protected from DNA digestion. Even the second peak and the third peak is visible, which represents a D and trinucleosome complex. Moreover, at the level of the larger marker peak, around 10,000 base pairs, larger DNA fragments are visible, which may be traced back to larger cfDNA molecules, published an example for inflammatory diseases or cancer. But this broader peak may also represent genomic DNA from leukocytes, which then would rather reflect a reduced plasma quality. It should also be considered that these size profiles only focus on the double-stranded DNA present in the ileate. Single-stranded DNA with a higher fragmented pattern, as revealed by specific libraries which focus on both double-stranded and single-stranded DNA, is not visible here, but has most likely a significant impact on the total cell-free DNA yield. This shall give you a first example of the bias each analysis or quantification method has, hardly avoidable due to the low concentrations of cell-free DNA, which makes direct and easy quantification not possible, and therefore results in only a limited number of analysis methods available. The lower right figures merge the cell-free DNA size profile generated from a urine in blue and from plasma in red, both from 4 ml sample input. For urine, next to the prominent peak at 170 base pairs, an additional peak with a broad range from 20 to 100 base pairs, interrupted by the lower marker peak, is visible. These 2 peaks may reflect the cell-free DNA fraction derived from plasma, highly fragmented during the glomerular transfer, and the fraction derived from the urinary system. Based on our knowledge, working with cell-free DNA from urine is most challenging, mainly due to the high need for stabilization to prevent the release of genomic DNA from cells present in urine, and in parallel to prevent the fast degradation of cell-free DNA in urine, which takes place immediately after sample collection within a few minutes. But usage of standardized workflow which address both issues makes urine to an easy and in high volume available and complete non-invasive sample material. A robust and sensitive quantification method for cell-free DNA is the PCR. We use for quantification a housekeeping gene, the multi-copy gene for 18S ribosomal DNA, amplifying a short amplicon of 66 base pairs to cover as much as possible from the fragmented cell-free DNA fraction. The figure here illustrates the high variability in cell-free DNA amounts, which shows, in this case here, a thousandfold difference from donor to donor in this experiment, including a sample size of 48 single donors. This illustrates that usage of an absolute quantification is not possible to evaluate the extraction efficiency for an individual sample. That's why we usually run a reference method to calculate the success of the extraction process. Here, the automated QIAsymphony circulating DNA kit on the x-axis was compared to the gold standard in the market, the manual QIAm circulating nucleic acid kit, shown on the y-axis. The 18S copies were plotted for each donor, which result in a comparable total cell-free DNA extraction efficiency for both kits for a broad range of cell-free DNA concentrations. In the last year, several providers from internal controls for cell-free DNA offer solutions to control the complete workflow, starting from the DNA extraction process via the downstream assay until the final data analysis. Despite the clear advantage to run for each sample an independent control, in particular due to the known challenges of the very low and variable cfDNA concentrations, it should be considered that these internal controls should be rather used qualitatively than quantitatively. That means they can give you a clear yes or no answer regarding the success of the workflow. But due to their expected bias concerning the extraction efficiency of the artificial DNA, depending on the used extraction kit or chemistry, the recovery of the artificial DNA must not mimic the extraction efficiency for the endogenous cell-free DNA for the used extraction kit. That's why we trust only on the results generated with real plasma samples and the corresponding endogenous cell-free DNA extraction. Compared to the previous slide, which focused on the total cell-free DNA yield, these data shall give you an example how to evaluate the extraction performance for low abundance genes. This aspect is most important because usually users are rather interested in the target cell-free DNA, that means the fetal or mutant biomarkers, which are present in only low ratios in the background of cell-free DNA of few percent or even lower. The upper figure here shows female plasma spiked with low amounts of male plasma to mimic a sample type with different ratios of a low abundant cell-free DNA in large background. By analyzing Y chromosome specific genes, the multi-copy gene DIS14 as well as the single copy gene SRY1. Using droplet digital PCR as readout, we first calculated a limit of detection because we had to consider a background noise of non-spiked female plasma here. Here we calculated a limit of detection indicated by the red line of 2 microliters male plasma diluted in 2 ml female plasma, shown on the x-axis, which corresponds to 7 copies DIS14 per PCR well on the y-axis. The lower right figures result from the same experimental design. That means the analysis of Y chromosome specific genes at the limit of detection in the normal cell-free DNA background, used as a model for a reliable detection of low abundance genes depending on the sample input volume. Female plasma was spiked again with male plasma from different plasma volumes, ranging here from 1 to 10 ml. To ensure a reliable detection of DIS14, female plasma was spiked with a 2 times limit of detection of DIS14 to minimize risk of dropouts for the low plasma input volume. The left figure on the upper right side for detection of DIS14 reveals a reliable extraction at the lower limit of detection and, in addition, a linear increase in yield for the elevated input volumes up to 10 ml plasma. The red circle here at 4 ml input shows a comparable extraction efficiency obtained to the reference, the QIAm circulating nucleic acid kit. Interestingly, when the same analytes were analyzed in parallel for the presence of the second low abundance gene, SRY1, which is shown on the right figure, the single copy gene SRY1 was analyzed below the calculated limit of detection. Therefore, only a hit rate study, that means the presence of droplets, was executed for the different input volumes. Using lower plasmid input volumes resulted in an unreliable detection of positive droplets in digital PCR, whereas sensitivity of the assay can be increased by using the highest input volume of 10 ml for the cell-free DNA extraction on the QIAsymphony. These data highlights once more the advantage of an increased sample input volume on the detectability of low abundance genes. Such an experimental setup is very close to the characteristics of clinical samples and therefore suitable for a sensitivity assessment of the complete workflow, which often focus on the sensitivity of the downstream assay. These data hopefully convince you that also the choice of the cell-free DNA extraction kit and the consideration of the required sample input volume plays a major role to achieve the needed overall workflow performance. I would like to finish my talk with a short summary. Circulating cell-free DNA is a new star arising in the field of molecular diagnostic with a huge potential for an early and straightforward detection of genetic aberrations present in example in pregnancies, cancer, or transplantation. To address the already existing but also the strong increasing demands for a reliable and automated cell-free DNA extraction process, combined with a high throughput, QIAGEN developed the QIAsymphony Circulating DNA Kit, well established for several years now in the market. Its pre-filled and barcoded reagent cartridges ensure a safe and ease of use in a diagnostic environment. The kit was verified for purification of cell-free DNA from human plasma and urine from 2 ml and 4 ml sample volume. It allows the generic use of plasma from non-stabilized as well as cell-free DNA stabilized blood collection tubes. And the dedicated cell-free DNA chemistry ensures an efficient extraction efficiency for highly fragmented DNA with a focus on low abundance DNA targets. Many thanks for your attention.